Other than being very fancy regeneration, actually it is very important in the bioassays. Okay, so bioassays is one of the important testing in pharmaceutical and biopharmaceutical industry, where it can help to find out what kind of cells are they stem cells and what kind of relationship that they have between the pharmaceuticals and the body cells. A lot of the people might have heard about stem cells, and stem cells, there are different types of stem cells. For those which is found in the bone marrow, in the umbilical cord blood and the peripheral blood, it is called a hematopoietic stem cells, and it is undergoing a procedure called haematopoiesis to become specialized and becoming all the blood cells that we have found in our body. And one of the important stem cells is called the mesenchymal stem cells, which is one of the hot research topic recently. It regulates the proliferation and differentiation of HSCs. So this is an example of blood cells that has been specialized from hematopoiesis, and they were from hematopoietic stem cells. The cells, they can be lymphocyte, natural killer cells, erythrocyte, basophil, eosinophil, neutrophils, monocytes or macrophages, and also basically most components of the blood cells. And it is important to be studied the hematopoiesis mechanism, and there are a lot of research is wanted to explain the HSC expansion and transplant, and there are some of the very important therapies such as cell-based therapies, and it also gives information about drug screening platform, where we can talk about it in the next slide. So HSCs, they are a very important component to form bone marrow models, and these models, they are very important because they can be mimicking the body situations, and there are a lot of 2D suspension cultures with cell layers and growth factor, and there are increasingly more 3D cultures using hydrogel, which is a polymer swollen with lots of water to mimic the condition in body with a lot of proteins and those microenvironment, the pH, the water content, and we try our best to have the comprehensive microphysiological systems such as MPS to replace the 3D culture models because they are more specific, and it can mimic the blood flow, shear stress, and the gradients of nutrients can be provided, and it will be like a really real cells. There are lots of sounds of anti-animal cruelty because a lot of testing they are run on animals prior to the human trials, and it is important to see how APIs interact with body, and if possible we can use this MPS to see if it can be accurate, and it can be a good choice because if it is a successful media, it can be produced in large scale and a lot of engineering can be done, and the analyzer, they can be done automatically, and there are a lot of high throughput screening, and it will save a lot of cost and time.